Welcome everyone to week number 5 in Science 200. We are past our halfway point now, so that is awesome. I had a bit of excitement this week. My iMac crashed on Wednesday night, and Thursday I went and bought myself a new one, and I have it all set up now, but yeah, there's all kinds of things going on. So we'll see how I do with this video this week. All right, so let's see what we've got happening. Last week was all about scientific communication, deciding who your audience is, deciding how you're going to communicate with your audience, how much technical jargon you can use, how many complicated graphs and tables you could use, what kind of language you're going to use. You anticipate the kinds of questions that your audience would ask you so that you would be prepared for this. So figuring out your audience and how you're going to present is important no matter whether you're doing this in science or in a business meeting or to your kids' class or whatever it is. So this was an important chapter for everyone. This week is all about biases in science and how we need to take out bias if we can figure out what it is and take it out so that the results that you get are the actual results that you get. So there are lots of different biases, like there's researcher bias. Like I could set up my experiments because I have an expectation of what things I think are going to happen. You can have confirmation bias, where I only look at the results that I have expected to get, and I sort of ignore the ones that I don't want to get. You have bias that your patients or your subjects or your clients, whatever you call them in your study, can bring. They know that you're expecting some type of result, so they may tell you that. They may indicate that these are the results that they are seeing. So there's all types of biases. It's important, if at all possible, to do double-blind studies. And when I participated in the 2 Pfizer COVID vaccine trials, they were clinical trials, neither the researchers, the doctors at the facility, the nurses, or I knew what kind of shot I was getting. The only person that knew was the one person who gave me the injection, and she wrote it down and put it into the computer, and nobody else had access to that information. That's a double-blind study where the researcher and the subject, neither one know who's getting which type of treatment. So those are really important, and most clinical trials are set up that way. We also talk about statistics when we analyze data in science, and we talk about p-values, and we talk about confidence levels. And they're sort of opposite from each other. So p-value is the probability, and you're looking for p-values less than 0.05%, of 0.05, a p-value. And so that's a probability of 5%. If you change that decimal into a percentage, it's 5%. And well, what the heck does that mean? That means that 5% of the time or less, this particular phenomenon is caused only by chance. So you want a high probability that the thing that is happening is really happening and not just being caused by chance. So you want p-value to be really, really low. On the other hand, confidence levels you want really, really high, because they give you confidence that the result that you're seeing is based on what you think is causing it. Okay? So we do statistical analyses so that we can show how this phenomenon is most likely to happen because of why we're saying it's happening. And that's based on lots and lots of statistical analyses, and you have to pick the right statistical test, and there are just all kinds of information out there about this. I had to take a graduate level class in statistics. It was the most horrible thing I ever took in my life, because I still don't know the difference between a one-tailed student t-test and I don't know. There's so many of them, ANOVA and all that. Anyway, you get it figured out when you need to analyze your data. We also want to mention Gregor Mendel's P experiments, which are talked about in Chapter 5 in the Sumo web text. This is a phenomenal study. So Gregor Mendel was an Austrian monk, and he was very interested in how traits are passed along from generation to generation. And the easiest thing that he could figure out to study was pea plants, because there were lots of variations in pea plants that farmers had access to. They produced different colored flowers, different colored leaves. They were different heights. The peas themselves were wrinkled or non-wrinkled. So they had all different types of traits. So Gregor Mendel planted some of these peas, and then he crossed some of them to see how the traits manifested themselves in the offspring. The most important thing that Gregor Mendel did was count. He counted the offspring. He recorded every offspring that he got and that he grew, and he knew who that cross came from. And then he recorded how many of those he got. And because of this, he figured out that there were some traits in the pea plants that would disappear in some generations, just be completely missing out of a generation. And then all of a sudden, when he continued to cross those plants with each other or other plants, he would see the return of those traits. And he came up with the two terms, dominant and recessive traits, and you all know those. We still use those terms today when we're talking about genetics. Did he understand anything about DNA? Absolutely not. Did he know what a gene was? Absolutely not. But he said that in each organism, there are 2 copies of each trait. Very important. We know that's true. We have 2 chromosomes of the same type. So we have 2 chromosome number 1s. One came from our mother, one came from our father. That's exactly what Gregor Mendel said back in the 1800s, before we even understood the molecule of heredity, DNA. So his experiments were instrumental in us understanding genetics on a basic level. You're also going to read some more about the Deepwater Horizon oil spill in 2010 and how we didn't count on just the BP scientists to give us information about this oil spill. The government had to step in because we were not able to shut off that oil spill. And one of the reasons was because the BP scientists were saying that about 1/10 the amount of oil was coming out of it that was actually coming out of it. So there was no way we would ever have capped that off if we used their statistics. So it's really important to remove bias. And this particular example is great for explaining this. The governmental scientists had no bias in this case. They had nothing to gain or lose from giving any information that they figured out, but the BP scientists certainly did. They wanted to minimize the impact of this particular oil spill. So it's a good study in having unbiased information. All right, this week you have no discussion board postings. You're going to revise your rough draft from week number 4. So I will grade those, and you will fix anything that you need to fix. Week 5 is really important because whatever you're writing in week 5 is what you're going to turn in week 6 after you've taken the rough edges off of it and fixed everything you need to fix. But fix what you can up until now. You're going to add the hypothesis section. So what in the heck? You already had a question that you had to submit in week 2. The question that you asked was a big overarching question. Like, are GMOs safe to consume as human beings? Or can genetic testing be done, genetic engineering be done, to repair diseased genes in embryos? How widespread is ocean acidification? is there harm being done to marine life because of ocean acidification, right? These are huge questions. And that's fine. That gave you a starting point for what to look for when you started doing your research. Your hypothesis is different. It's a much more focused question that you could actually test, right? So if you're talking about a big issue like ocean acidification, you could pull ocean acidification into the lab and test the effects of different pH levels on, let's see, the polyps of jellyfish, newly hatched polyps of jellyfish, right? You could take them into the lab and you can test the effects of pH on those. You could test the effect of lowering pH, changing pH, in fact, on calcium in the shells of shellfish, something like that. So your hypothesis is something testable, it's much more focused, and it's much more specific. And then you're going to explain to us how a scientist would go about answering that particular question. You can either put it in the form of a question, or you can put it in the form of an if-then statement. Either one's okay with me. Use your rubric and your guidelines to guide your work. That's very important. And tip of the week, use your feedback from me to improve your work. So every week I'm grading things and I'm taking off points if you're not doing something right, and I explain that in the feedback, what is going on, what I expect this section to look like, how I think you could improve this section. So use that feedback from me to improve your work, and you want to do this before you get to week 6. The thing is, every week you've been doing assignments that are building, the introduction, the question, right, the sources, everything that you did last week with the natural principles in your audience and that sort of thing. Everything is building until you get to week 6. In week 6 you're turning in a paper, a finalized paper of your particular topic, and it counts for a whole lot more points than all these other assignments have. And so that's why you want to improve everything that you have turned in so far so that you will get the maximum number of points in week 6. And then you say, well, aren't I finished then with my topic if I turn in my paper? No, because you're going to make a presentation about your topic. The paper is to prepare you by helping you do the research that you need to do and figuring out questions to ask and all of that before you put together a presentation. Week number 7, you're putting together speaker's notes, everything you would say to your audience. And week number 8, you're putting together a slide presentation, 5 to 7 slides, that you would show to your audience. You've already chosen who they are. You would show to your audience to explain your topic to them. So you got a presentation to make in weeks 7 and 8. And you've got some discussion boards coming up in week 6 and week 8. So that's what's coming up. Use your feedback from me to improve your work. If you need any help, if you have any questions this week, if you are totally stuck on that hypothesis thing, please just email me and let me know. I am glad to help you. That's what I'm here for. I will help you any way that I can. So just reach out and ask me for help. With that being said, I hope that you have a wonderful week number 5.